Welcome, my dear students. Today we will talk about alpha helix. The alpha helix is one of the most important and common secondary structures found in proteins. The structure was first proposed by Pauling and Corey. This helical arrangement of the polypeptide chain is stabilized mainly by hydrogen bonds. Its elegant right-handed twist and regular repeating pattern make it a key structural features in many proteins, from keratin in our hair to enzymes in our cells. Understanding the alpha helix helps students grasp how protein shape determines protein function, a concept essential for undergraduate, postgraduate, and NET examinations. Pauling and Corey firstly proposed the alpha helix structure. They understood the importance of hydrogen bonds in arranging the peptide bonds. William Astbury's X-ray studies 1930s showed alpha keratin hair quills has a repeating structure of 5.15 to 5.2 angstrom. What is the basic structure of the alpha helix? It's a right-handed helix. Its backbone is wound tightly around an imaginary central axis. The R groups project outwards. One turn of the helix length is 5.4 angstrom. It contains roughly 3.6 amino acids. Common structure in proteins, about 25% of amino acids are in alpha helices. Why a alpha helix forms easily? It forms maximum internal hydrogen bonds. Hydrogen bond occurs between C double bond O of residue of one residue and NH of residue of N plus 4 or I plus 4. So each turn is held by 3 to 4 hydrogen bonds, giving strong stability. Stereochemistry: helix can form all L amino acids or all D amino acids but not mixed. Natural proteins have L amino acids, only right-handed alpha helix is common. Factors affecting alpha helix stability: electrostatic interactions, that is the charge effect, destabilizing effect. Many glutamic acid which are negatively charged, when close together they repel each other and it destabilizes the alpha helix. Many lysine or arginine which are positively charged amino acid, when come close together they repel each other and it destabilizes the alpha helix. And stabilizing: oppositely charged residues at position I and I plus 3 or I plus 4 attract each other and it stabilizes the alpha helix. Bulkiness, that is size and shape of the R group. Asparagine, serine, threonine, cysteine come close together, and as they are too bulky they destabilize the alpha helix. Interactions between residues which are 3 to 4 apart. The helix geometry brings residues I and I plus 3 or I plus 4 close, and it can form ion pairs, that is oppositely charged residues, and it can show aromatic stacking with the help of the amino acids like phenylalanine, tyrosine, tryptophan, etc. Special amino acids such as proline, which is called as helix broker, it causes a kink because rigid ring structure, there is no rotation around the NC alpha bond, so it lacks NH and it cannot form hydrogen bond. It is rarely found in a alpha helix. Glycine is too flexible, helix becomes unstable and often disrupts the alpha helix. Helix dipole effect or end effect: helix has a net dipole, that is N terminus is always partial positive and C terminus is always partial negative. Stabilizing locations: near N terminus, negatively charged residues such as glutamic acid and aspartic acids are found. Near C terminus, positively charged residues such as lysine, arginines are located. Destabilizing locations: wrong charges near ends can destabilize the alpha helix. So summary: the five constraints affecting the alpha helix stability: charge repulsion or attraction between nearby residues, bulkiness of adjacent R groups, interactions between residues I and I plus 3 or I plus 4, presence of proline and glycine, helix dipole interaction at N and C termini. So to sum up, we can say that the alpha helix beautifully demonstrates how simple interactions like hydrogen bonding can create a stable functional structure within proteins. Its stability depends on amino acid sequences, side chain interactions and helix dipole effects. By mastering the features and formation of the alpha helix, students build a strong foundation for understanding higher level of protein architecture and answering advanced exam questions effectively. Keep revising the examples and ratios because these topics are absolute favorites in competitive exams. See you in the next class. Thank you.